everyone, my name is DAVAA. I studies at National University of Mongolia. I will present my research on soil geographic diversity and its relationship with soil organic carbon and vegetation cover. This is the outline of my presentation. Soil is very important terrestrial ecosystems. Soil geographies help with the nutrient cycling, decomposition, and soil formation. The Gobi ecosystem covers more than 25% of Mongolia. It's very sensitive to changes in temperature and precipitation. Therefore, studying soil biodiversity in this region is important. E-DNA metabolic coding is widely used to study soil biodiversity. Previous studies show that vegetation cover and soil organic carbon affect soil biodiversity. The aim of this study was to use eDNA to study soil geographic diversity. We also looked at its relationship with soil organic carbon and vegetation cover. This study was carried out in Bayongur province, Mongolia. We collected 20 soil samples from desert steppe and desert ecosystems. At each site, we collected 5 soil samples. Then we mixed them into one sample. The samples were preserved with silica gel, and DNA extraction and sequencing were done at Spigen laboratory in France using the 18S RNA marker. Soil samples were collected for soil organic carbon analysis. The samples were air-dried and sieved before laboratory analysis. Vegetation cover was measured using a 1x1 meter contract. Soil organic carbon was measured using the loss on ignition method. Then soil organic carbon was calculated. Using eDNA metabolic coding, we detected a total of 162 eukaryotic taxa. Fungi were the dominant group, followed by animals, horomista, and plants. We found 3 community groups. The ANOS-IM test showed significant differences among the groups. However, the silhouette index was low, so the groups were not clearly separated. We found significant indicator taxa in cluster 1 and cluster 3. We did not find any significant indicator taxa in cluster 2. Effect of soil organic carbon and vegetation cover on community structure. Soil organic carbon alone was not significant. Vegetation cover alone was also not significant. However, together they had a significant effect on community structure. This graph shows the relationship between vegetation cover and channel diversity. As vegetation cover increases, channel diversity also increases. This relationship was statistically significant. Overall, we detected 162 eukaryotic taxa. We found 3 significant community groups. Vegetation cover had a positive relationship with channel diversity. However, soil organic carbon, soil moisture, and plant biomass were not significant. Fungi were the largest group. This is similar to previous studies. We found significant indicator taxa in cluster 1 and cluster 3. Primary bias may have affected the detection of some taxa. In conclusion, vegetation cover was the main factor related to soil geographic diversity. Community structure was affected by vegetation cover and soil carbon together. These are the differences used in this study. Thank you for your attention.